Hello and welcome to the next tutorial video. And in this one we're going to be talking about the energy systems, but we're going to be talking about them in slightly different terms. So far when we've been talking about the energy systems, we've always been assuming that our initial fuel is glucose or carbohydrate. And this isn't always the case, as we know we can get fuel from different macronutrients in our body, such as fatty acids and fats, or indeed proteins. Now, most of the time proteins are not used very much for providing fuel. They have lots of other jobs in the body, and there's not really any large stores of proteins in the body. When they're ingested, they tend to get used up and metabolized very quickly. What happens here is the nitrogen is removed from the amino acids and they get reconfigured into glucose or fat. But this only really happens in extreme circumstances. So if somebody's suffering from malnutrition, or there's just a complete absence of fats or carbohydrates in the body. This is an example of the process of gluconeogenesis. So neo meaning that glucose is produced but from a non-sugar based source. So that's all we're really going to say about proteins because it doesn't happen very often. Mostly we're going to be talking about the use of fatty acids as an other form of fuel. This is what the overall breakdown of fats looks like. So fats, as we know, are stored in the adipose tissue as triglycerides. And they're broken down in a process called lipolysis into fatty acids and glycerol. The glycerol head can then be taken off to the glycolysis process, whilst the fatty acids are transferred in the blood to the muscle. Once in the muscle, they're broken down into acetyl-CoA by a process of beta oxidation. And hopefully you'll recognise acetyl-CoA. So acetyl-CoA is used as the entry point of the Krebs cycle. When fats are broken down, the glycerol head gets used in glycolysis and the fatty acids get transferred into acetyl-CoA. We have two breakdowns here. We've got lipolysis, where the fats are broken down into glycerol and the fatty acids. And we've also got beta oxidation, where those individual fatty acids are broken down into acetyl-CoA. Now, the breakdown of fats produces huge amounts more energy than the breakdown of carbohydrates. This is why they're seen as a very high energy store. What we're going to do is have a look at exactly why it produces so much ATP. You don't need to know this for the exam, but it's quite interesting to look at and try and realise why fats produce so much more energy. Let's remind ourselves what a fat looks like. A fat is a triglyceride molecule. We've got our glycerol head on the left and then 3 fatty acid chains. And here's an example of one of those fatty acid chains. This is palmitic acid. As you can see, it's a 16 carbon chain and it's quite commonly found in certain parts of the diet. And we're going to be specifically using this as an example. In the process of lipolysis, the triglyceride is hydrolyzed to release the glycerol head and the 3 fatty acids. The glycerol head gets used in glycolysis to form pyruvate, and the fatty acid chains are transformed into acetyl-CoA via a process called beta oxidation. That acetyl-CoA can obviously then be used in the Krebs cycle. Just a quick note, for this to occur, oxygen must be present. If you're not working aerobically, this process cannot take place. Let's look at it in a little bit more detail and just start seeing how much energy is produced. Palmitic acid is a 16 carbon fatty acid. It's commonly found in butter, cheese, milk and meat. As a 16 carbon fatty acid, it goes through a process of beta oxidation 7 times. Now what we mean by that is that each 2 carbons get broken off to form 1 acetyl-CoA. If we were chopping up our chain of palmitic acid with 16 carbons, we would have to make 7 cuts in order to separate it into 2 carbon molecules. That means it goes through the process of beta oxidation 7 times. Each time it does that, it breaks off 2 carbons. They then get changed into acetyl-CoA. but it also produces NADH and an FADH. Overall, the process of beta oxidation produces 7 NADH, 7 FADH, and 8 acetyl-CoAs. Here we have our first 2-carbon molecule being broken off, and then the next one, and the next one. And this continues until the entire chain is broken down into 2-carbon molecules, which can then be transferred into acetyl-CoA. Let's look at what happens to the acetyl-CoA now when that goes through to the Krebs cycle. Each acetyl-CoA, as we know from looking at glucose as a substrate, each acetyl-CoA produces 3 NADH, 1 FADH, and 1 ATP in the Krebs cycle. Overall then, the 8 acetyl-CoAs will produce 24 NADH, 8 FADH, and 8 ATP. Still with me? Okay, what we need to do is we need to add on the NADH and FADH that were produced in beta oxidation. So that accounts for an additional 7 NADH and 7 FADH too. So overall, the breaking down of that palmitic acid chain produces 31 NADH, 15 FADH2, and 8 ATP. Obviously, our electron carriers then go forward to the electron transport chain just as they would as if glucose was used as the fuel source. And the same thing occurs, so the NADH is able to resynthesize 3 ATP molecules, and the FADH is capable of resynthesizing 2 ATP molecules. So overall, 31 NADH is worth 93 ATPs. 15 FADH2s are worth 30 ATP, and then we can add in the 8 ATP molecules which are produced in the Krebs cycle. So in terms of the ATP that we get from breaking down 16 carbon chain of palmitic acid, we end up with 131 ATPs. Now this seems quite a lot in itself, but don't forget that we're talking about a triglyceride. So there's 3 fatty acid chains. Let's assume for a minute that we've got we've got 3 fatty acid chains which are all exactly the same. There's going to be 3 chains of 131 ATP. That's going to give us 393 ATP molecules from 1 triglyceride. And that's not even considering the amount of energy that's produced by the glycerol head going through to form pyruvate in glycolysis. As you can see, this is a huge amount of energy compared to the breakdown of a single glucose molecule. If you'd like to take the test on the use of fats as an energy substrate, please click on the link here. Thanks very much.